Chapter 13 Photosynthesis. Dark reaction or biosynthetic phases. Use of ATP and NADPH. The products of light reaction are ATP, NADPH, and O2. Of these, O2 diffuse out of the chloroplast while ATP and NADPH are used to drive the processes leading to their synthesis of food more accurate. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the products of light reaction, that is ATP and NADPH, besides CO2 and H2O. It can be verified as follows. Immediately after light becomes unavailable, the biosynthetic phase process continue for some time and then stops. If then light is made available, the synthesis starts again. So it can be said that biosynthetic phase is dependent on the product of light reaction. So it is also called as the dark reaction. In dark reaction, carbon dioxide when react with a water molecule in presence of sunlight and chlorophyll C6H12O6 plus O2 plus H2O is produced. This reaction is of photosynthesis, right? If we just see the reversible process that when the sugar or glucose molecule is break down in carbon dioxide and water molecule, respiration takes place. We can say that photosynthesis and respiration are opposite or reverse of each other. Moreover, in photosynthesis, when the water molecule is photolyzed or breakdown of water molecule take place, oxygen is released. This reaction is known as light reaction or oxidation phase. While when carbon dioxide is used to produce sugar in form of glucose, this reaction is known as a dark reaction or reduction phase. In photosynthesis, energy is used, while in respiration, energy is released. Dark reaction. Carbon dioxide is combined with water molecule to produce sugar or CH2O whole N. For that, ATP and NADPH are used. It was of the interest to scientists to find out how these reactions proceed, or rather, what was the first product formed when carbon dioxide is taken into reaction or fixed. Also, after the World War II, among the several efforts to put radioisotope to beneficial use, carbon dioxide assimilation during photosynthesis is of two types: C3 pathway and C4 pathway. The use of radioactive C14 by Melvin Calvin in algal photosynthesis studied lead to the discovery that the first carbon dioxide fixation product was 3-carbon organic acid. He also contributed to working out the complete biosynthetic phase. The first product was phosphoglyceraldehyde (PGM). Moreover, the alga which was used for his study was of chlorella. In 1950s he has studied. With Melvin Calvin, another scientist was there named A. Balson and their colleagues were there. So the cycle is also known as Calvin-Benson cycle. As reduction is taking place, this cycle is also called reductive pentose phosphate cycle. Hence, it is also called Calvin cycle. The first product identified was 3-phosphoglyceric acid, or in short PGA. Another cycle was C4 pathway. In this, first stable product is oxaloacetic acid OAA of 4-carbon acid. Scientists conducted wide range of experiments over a plant, lead to the discovery of another group of plant with a first stable product. This acid was identified to be oxaloacetic acid. It is also known as a Hertzsprung pathway or dicarboxylic acid pathway. Since CO2 assimilation during photosynthesis was said, these two main types were found. Besides this C3 and C4 pathway, another cycle was there which was known as CAM cycle, CAM, which stands for crassulacean acid metabolism cycle. It occurs mainly in dicot plants. The study very unexpectedly showed that the acceptor molecule was a 5-carbon keto sugar. It was ribulose biphosphate, RUBP. They also believed that since the first product was a C3 acid, the primary acceptor would be 2-carbon compound. They spent many years trying to identify 2-carbon compound before they discovered the 5-carbon RUBP. It occurs, C3 pathway occurs in all photosynthetic plants. It has 3 stages: carboxylation, reduction, and regeneration. It does not matter whether they have C3 or C4 or any other pathway. Here is the C3 cycle in which all the 3 processes are taking place. Carboxylation in this RUBP was converted to 3-PGA in presence of enzyme named rubisco. From phosphoglyceric aldehyde, it is converted to triose phosphate, that is 3 carbon molecules. That process was known as reduction, and again triose phosphate is converted or regeneration takes place of RUBP. In this way, whole cycle takes place. Here various reactions are taking place in presence of different type of enzymes, such as in carboxylation, carboxylase is taking place to convert into 2-carboxy 3-keto 1,5-biphosphorybatol. In second, it is also known as a reduction phase, is also known as a glycolytic reversal. In this, phosphoglycerate kinase is taking place to convert PGA into 1,3-biphosphoglyceric acid. And further, in the presence of various or with the help of various reactions and in presence of various enzymes, it has been converted into glucose molecule. But fructose reaction takes place convert in 5-carbon molecule which is known as a xylose, and that process takes place for the regeneration of RUBP with the various type of reactions. It has been converted in the presence of different phosphoribulose into ribulose 1,5-biphosphate. In this way, whole cycle is taking place. If we just see the various steps, first step takes place carboxylation of RUBP. In carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of Calvin cycle where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which result in the formation of two molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco. Rubisco is the most abundant enzyme in the world. It is one type of protein only. Dark reaction. Carbon dioxide is combined with water molecule to produce sugar or CH2O whole N. For that, ATP and NADPH are used. It was of the interest to scientists to find out how these reactions proceed, or rather, what was the first product formed when carbon dioxide is taken into reaction or fixed. Also, after the World War II, among the several efforts to put radioisotope to beneficial use. Carbon dioxide assimilation during photosynthesis is of two types, C3 pathway and C4 pathway. The use of radioactive C14 by Melvin Calvin in algal photosynthesis studied. Third step is regeneration of RUBP. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps required 1 ATP for phosphorylation to form RUBP. It can be denoted in the equation form PGAL in the presence of ATP produce RUBP. Hence, for every carbon dioxide molecule entering the Kelvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. Here is the C3 cycle in which all the three processes are taking place. Carboxylation in this RUBP was converted to 3-PGA in presence of enzyme named Rubisco. From phosphoglyceric aldehyde, it is converted to triose phosphate, that is 3 carbon molecules. That process was known as reduction, and again triose phosphate is converted or regeneration takes place of RUBP. In this way, whole cycle takes place. Here various reactions are taking place in presence of different type of enzymes such as in carboxylation, carboxylase is taking place to convert into 2-carboxy 3-keto 1,5 biphosphorybatol. In second, it is also known as a reduction phase is also known as a glycolytic reversal. In this phosphor, now one can think that where does this sugar used or glucose is used? This glucose which is produced inside the C3 cycle is used in the formation of sucrose. Sucrose is nothing but a disaccharide or mixture of glucose plus fructose, which is responsible for the transportation of carbohydrate in plants. Another, it is used for the starch. Starch is nothing but polysaccharide, a glucose polymer, long chain of glucose, which is used for the storage of the carbohydrate in different parts of the plant. Cellulose, it is also one type of byproduct of glucose. It is present in the cell wall of plant and responsible to give the structure to the cell wall of the plant. It is also used for fatty acid plus glycerol to store the oil in the plants. Another thing, they are responsible for the amino acids, and amino acids are responsible for the protein synthesis. In this way, glucose is used for various formations